Now we're at Unit 8, Eukaryotic cells. We're going to look more specifically at eukaryotic cells, look at the types of organelles that they have within them. And the first part here is going to be focusing on animal cells. We'll look at plant cells more specifically in Part B. The chapters that you want to be referring to, if you wish to refer to them, in the book are as follows. It's a little bit cut up here, 3.1, 3.2, 3.4a, 3.4b, 3.5, and 3.6. Later on we will focus on 3.3 and 3.4, especially 3.4c there in Part B. So we are going to be looking at, we're going to be looking inside the eukaryotic cell. We're going to look at all of the various structures within, all of the various organelles. The nucleus, the endoplasmic reticulum, the Golgi apparatus, the mitochondria, so on and so forth. We'll look at what these things are, what they do, what they're called, and so forth. Now as we look at these various organelles, remember organelle means a small unit of organization, we want to be thinking about them in terms of their structure, their chemical composition. What are they made out of? Are they made out of phospholipids? Are they made out of proteins? Or do they have enzymes within? What do they do? What is their function? The word function means what it does, what its practical purpose is. The relative size of the organelles to one another, and other tidbits of information that I will be sharing as we go through here. So that's what you want to be focusing on when you're taking down notes. You want to be looking at structure, chemical composition, function, relative size, and so forth. So we're looking at eukaryotic cells, so we're going to go ahead and look at that major feature of the eukaryotes, and that is the nucleus. The eukaryotes are distinguished from the prokaryotes by having this large membranous structure that holds the vast majority of the DNA that you're going to find in a eukaryotic cell. Remember that in the prokaryotes there is no such thing. They lack a nucleus. They have simply this nucleoid region where the DNA is just suspended inside the cytoplasm. So in the eukaryotes you have this large membraned organelle that's holding the DNA. And actually the nucleus is one of three organelles that we'll be looking at that is actually double membraned. It has a double membrane structure, one membrane on the outside and another membrane on the inside. And why is it double membraned? And there could be many explanations. One is that it's double membraned because it's trying to protect the DNA from damage. You don't want things from the cytoplasm, from the outside world, getting inside the nucleus and harming the DNA. If you harm the DNA is essentially the operating program for the cell. If the operating program goes down, then the cell goes down. So the double membraned nucleus is holding and protecting the DNA. Now, you might have been told at some point in time that you have 46 chromosomes in every cell in your body. It's quite possible that the book would make a statement like that. That is simply not true. You don't have chromosomes in most of your cells. You have DNA, but you do not have chromosomes. Most of the time, the DNA is not in chromosome form. Most of the time, it is in a form that is known as chromatin. Most of the time, your DNA is in a long, loose, strand-like formation. It's oftentimes referred to as being like a bowl of spaghetti noodles, these DNA molecules inside the nucleus. Well, when it's in this long, strand-like condition, it's referred to as chromatin. The word chromatin literally means dark stuff. At a certain point in the life cycle of a given cell, the chromatin, the DNA, will be doubled up. There'll be twice as much of it. This happens when the cell is going to be dividing. It's about ready to divide, so it doubles up the DNA so you can have two copies of the DNA, one for one resulting cell and one for the other resulting cell. So you double up the DNA, you double up the chromatin, and then the cells go ahead and they condense the chromatin down. They condense it down to a chromosome. Chromosome means colored body. So the chromosomes only appear once in a while you are in the process of replicating the cells. In the meantime, it's always in chromatin form. So the nucleus is holding the DNA in chromatin form. What is I mentioned the word chromatin means dark stuff. Why would it be called that? Well, back in the early days of microscope science, back in the late 1700s, early 1800s, the various microscope workers are looking through their microscopes, and they're seeing this dark stuff. They had stained the cells with various kinds of dyes, and they can now see this dark stuff inside the nucleus. As it turns out, the DNA happens to take certain kinds of stains. And so everybody's writing to one another about this dark stuff that they're seeing in the nucleus, but the scientists, they might speak French, they might speak English, Dutch, Arabic, Norwegian, whatever it might be. So they're trying to communicate with one another. So they use Greek terms, Greek and Latin, and they're seeing this dark stuff inside the nucleus, and they're calling it chromatin, which simply means dark stuff. Now the other feature inside the nucleus there is that dark mass, that dark ball right there. It's referred to as the nucleolus, which literally means the little nucleus, but it's not a little nucleus. It doesn't have any membrane structure around it. It's just an area of DNA that's somewhere kind of halfway between chromatin and chromosome form. It's a little bit condensed, but not condensed all the way. The nucleolus has the distinction of being that area of the nucleus that holds the DNA that has the information for how to make the ribosomes. And they think that this might be the oldest portion of the nucleus, this area that manufactures or holds the information for how to make the materials for the ribosomes. The materials are assembled outside of the nucleus, but the nucleolus holds the information for how to make the rRNA, the ribosomal RNA, and the proteins that are associated with the ribosomes. Now the DNA is in the business of holding information for how to make polypeptides, and in the business of manufacturing messenger RNA, mRNA, that will go ahead and tell the ribosomes what polypeptides to make. But here's the deal, there are no ribosomes inside the nucleus. All the ribosomes are outside the nucleus, so how are you going to get the messenger RNA information out of the nucleus? And the answer is the nucleus is equipped with these pores, or pore complexes. In fact, if you look at the outside of the nucleus, you see it kind of looks like the surface of the moon. It's and these craters are pores. They're actually made out of polypeptides themselves, a bunch of polypeptides put together. And they are embedded, sort of like rivets, into the double membrane structure of the nucleus, what they call collectively the nuclear envelope. A bunch of polypeptides, 8 polypeptides put together in a ring, and they are referred to as nuclear pores. Now here's an illustration of a nuclear pore, and if you look at it, it doesn't really look too much like a pore. It looks more like a filter, and in fact that would be a better word for it. It's allowing certain things in or out of the nucleus. One of these is the mRNA. It allows the mRNA to leave the nucleus and go on out and communicate with the ribosomes. Also, there are certain things that you might want to have come into the nucleus. So the nucleus, the DNA, does in fact need information as to what's going on inside the cytoplasm. If, for instance, let us say there's a large amount of a certain type of sugar that needs to be broken down, well, either that sugar or residues from that sugar will enter into the nucleus and activate various genes that might now start producing enzymes that will break down that sugar. At a certain point in time, in human beings, let's say the human male, testosterone begins to be produced in large amounts, and the testosterone will, in combination with other materials, enter into the nucleus and activate various genes that will do things like thicken up the vocal cords, increase muscle mass, bone density, things of that sort. Now, how does the testosterone get in there? It gets in there through the nuclear pore. So the nuclear pores are allowing things in. They're also allowing things out, again, specifically messenger RNA. The messenger RNA will be manufactured by the DNA in chromatin form, and now that that mRNA will leave. The messenger RNA is actually it's manufactured using the electromagnetic sequences on the nucleotides. So the DNA will be opened up, and an RNA, and specifically an mRNA, will be manufactured using the DNA template. And now that that mRNA will leave through the nuclear pore. Now, if it leaves through a nuclear pore like the one that we're seeing right here, it'll enter directly into the cytoplasm, and it will now go off and find ribosomes and feed the information into the ribosomes. On the other hand, it might leave through a nuclear pore like this one right here, and it will now enter directly into the endoplasmic reticulum, and we'll see that in just a bit. Here's an illustration of the nucleus there, and then the cytoplasm there in blue. And you can see the DNA in red there, looks kind of like a railroad trestle, opened up, and the mRNA is being manufactured. The mRNA looks like a little yellow centipede. And that process of manufacturing the mRNA is known as transcription. Now, we'll look at this in much more detail when we look at protein synthesis later on. But that little yellow centipede, the mRNA molecule, will exit through that pore and go out to the ribosome, where it will now be translated. Translation will take place out by the ribosome. The mRNA is being moved through the ribosome. The ribosome is using that information to take amino acids, the building blocks of proteins, and put them together into just the right sequence. That manufacturing of the protein using the mRNA as a messenger, as a source of information, is known as translation. And again, when we look at protein synthesis, we will look at both transcription and translation in a fair amount of detail. Now, consider the situation in which the mRNA is moving out through this kind of pore. Well, it's moving out into that sort of curtainous band of membranes that you see out there. A series of membranes just sort of suspended there, at least it seems to be suspended, out in the cytoplasm. Well, this organelle, this large, sort of curtainous series of membranes, is known as the endoplasmic reticulum. This is the longest name for any organelle, endoplasmic reticulum. The word reticulum means network. And endo means within. We'll see that word pop up here and there. And plasmic, again, it means stuff. So it means the network stuff inside the cell, I guess is what it's supposed to mean. Endoplasmic reticulum. There's actually two different kinds of endoplasmic reticulum. There's the smooth endoplasmic reticulum. We'll look at that later. And then there's the rough endoplasmic reticulum, which is what we're looking at right now. The difference between the two is that this particular series of membranous curtains, this endoplasmic reticulum, is studded with ribosomes. Sometimes people remember it not as the rough endoplasmic reticulum, but as the ribosomal endoplasmic reticulum, and it's abbreviated RER. Well, it's holding a bunch of ribosomes in place. Here's an electron microscope photograph of the endoplasmic reticulum. The nucleus is down in the lower left. And then you can see this series of curtainous membranes moving away from the nucleus here. And they have kind of a grainy or rough appearance. Well, that's because of all of the ribosomes that are studded on the surface. So when they first looked at it, they didn't know that it was ribosomes causing this grainy appearance, this rough appearance, so they just called it the rough endoplasmic reticulum. Well, why do you have this endoplasmic reticulum snug up against the nucleus? Well, here's the deal. When the mRNA leaves the nucleus, moves out through that pore, and moves into the endoplasmic reticulum, it is immediately surrounded by a bunch of ribosomes. It is in a target-rich environment. It cannot help but find a ribosome. Now, again, the mRNA can leave through different pores and never see the endoplasmic reticulum. Why would it go one way or another? Well, as it turns out, mRNAs that are coding for proteins that are going to be used inside the cell, they will go out into the cytoplasm in general. But ones that are coding for ones that are for proteins that are leaving the cell, they tend to go out into the endoplasmic reticulum. They bond to a ribosome there, and then they connect to the endoplasmic reticulum membrane. And we'll see why that is right about now. So you can see an mRNA there inside the endoplasmic reticulum. What is it doing in there? Well, it's feeding its information into ribosomes that are studded up against the membrane of the endoplasmic reticulum. And as these ribosomes take the information in, they use the information to put amino acids into a polypeptide. And then they take those polypeptides and they are actually feeding them into the interior of the endoplasmic reticulum, into the little space between the two membrane surfaces. So you get an mRNA that is released. It'll bond onto a ribosome. That ribosome will now head down towards the membrane of the endoplasmic reticulum. It will implant there, and now it will start to manufacture the protein. It might already have begun the process, but it continues that process, and it now inserts that protein. Actually, I should be saying polypeptide. It's not necessarily a protein yet. It's going to insert that polypeptide into that space of the endoplasmic reticulum known as the lumen in between the two membrane surfaces. So the polypeptides are being stored up inside the endoplasmic reticulum. What's going to happen here now is that bunch of membrane there with the polypeptides is actually going to pinch off from the endoplasmic reticulum, like so. So you have this little bubble there with the polypeptides in it. Well, this is now called a transport vesicle. A vesicle is a word that is used to refer to a small vessel. So it is a vessel. It's like a little ship. It's like a little boat. And it has these polypeptides inside of it. Well, it's going to transport the polypeptides off to a new destination, and that destination happens to be the Golgi body, or the Golgi apparatus, as it is called, these craters. How are the transport vesicles pinched off? Well, there's other proteins that come in and basically form a cage around it and pinch it off by using this cage. Now you have this transport vesicle that is now floating loose in the cytoplasm. And there's that transport vesicle right there. Now I said that it's floating loose in the cytoplasm. That's actually not true. It's actually on a track, a guided track that will carry it off towards the Golgi body, and we'll see how that guided track works later on. So there's that transport vesicle, and it's heading off toward the Golgi body, also known as the Golgi apparatus. Well, the Golgi apparatus is another bunch of membranes. The endoplasmic reticulum, its structure can be described as a series of curtainous membranes. Here you have sort of a collection of very thin, flat membranes, and they have a bunch of enzymes within. And these transport vesicles are bringing in these polypeptides, and these polypeptides, they're not proteins. Remember that a protein is a polypeptide that has a function. Any string of amino acids is a polypeptide, but it may not have any function. When the polypeptides are manufactured off the ribosome, they may not be ready yet. They might be immature. They might have to have things that are taken away from them or added to or cut into or have things spliced together. That's not what the ribosomes do. Some polypeptides, in order to be functional proteins, you might have to have an atom of zinc added to it. Well, the ribosomes don't deal with zinc. They deal with amino acids. So if you want zinc put on it, you're going to have to talk to somebody else. And that somebody else is going to be the enzymes inside the Golgi body. They're going to put zinc onto the polypeptide or iron onto the hemoglobin molecule, whatever it might be. They are remanufacturing the polypeptides that are coming in on the transport vesicles. Here's another view of this Golgi body, or now it's called the Golgi apparatus. Now it looks like bubble gum, a bunch of flattened membranes here holding a bunch of enzymes. And you can see the incoming transport vesicle right there. Coming in on the cis face, remember that cis means same, so it's the same side as the endoplasmic reticulum. And this incoming transport vesicle will release its polypeptides, and they will move through the Golgi apparatus, and they will be remanufactured by various enzymes, and when they're done, they're then packaged up into secretory vesicles. Now, the book refers to secretory vesicles as simply transport vesicles. I guess technically you can say that the secretory vesicle is a kind of transport vesicle, but most specifically, the secretory vesicle is holding finished proteins. The incoming transport vesicle has unfinished polypeptides. The secretory vesicle is holding finished proteins. And what are we going to do with these finished proteins? Well, the secretory vesicles are going to leave the Golgi apparatus, the Golgi body, and it is going to, it's going to release the proteins into the outside world. And there it is, releasing the proteins into the outside world. Well, that's why we call it a secretory vesicle, because it secretes the finished proteins to the outside world. You secrete sweat. Well, what that means to secrete something is that you're releasing it for a purpose. The sweat helps cool down the body. It also helps to nourish bacterial populations on your skin. When you excrete something, like urine, for instance, you're just getting rid of it. You have no further use for it. It has no purpose outside the body. So you excrete urine, but you secrete sweat. Well, these secretory vesicles are secreting these finished proteins. Now consider a protein like insulin, which helps to regulate blood sugar levels. That gene for insulin is found within the DNA. An mRNA molecule carrying the electromagnetic information for insulin is now manufactured. It moves out into the endoplasmic reticulum. The insulin protein is manufactured, but actually it's not finished yet. It is a pre-insulin polypeptide, and it is put into the transport vesicles, sent off to the Golgi body. The Golgi body rearranges the insulin pre-insulin polypeptide into real insulin, and now it puts it into secretory vesicles, and now it can be released. So that's the whole business of the Golgi body. It's remanufacturing polypeptides and then allowing them to be released to the outside world. So you have this whole flow of information that's going on inside the cells. You have that mRNA molecule, which is entering into the endoplasmic reticulum. The polypeptides are being manufactured and being stored up inside the endoplasmic reticulum. The transport vesicles butt off here, and they're carrying these polypeptides with them, and they release them into the Golgi apparatus. The Golgi apparatus works on them and puts them into secretory vesicles, and now releases them to the outside world. So you have this whole flow of information from the nucleus to the rough endoplasmic reticulum, the transport vesicles, Golgi apparatus, secretory vesicles, and then out to the plasma membrane. It's referred to as the endomembrane system. This system of membranes that are all working together, this whole flow of information and material from the nucleus to the outside of the cell. Endomembrane system, the system of membranes inside of the cell, the interior membrane system. So there you have it, a whole bunch of organelles all working together for a common purpose. There's other organelles that are not part of the endomembrane system that are just as important. One of them is the lysosome. This little bluish bubble there in the corner of the cell. The lysosome may or may not be part of the endomembrane system. Some people believe that they are manufactured by the Golgi apparatus, so that might make them part of the endomembrane system, but we don't really need to worry about that. What are these lysosomes? Well, they're actually small vesicles, but we don't call them vesicles. We call them lysosomes. Lyso means to split, and some means body, so they are splitting bodies. They contain enzymes within that can break down a wide variety of organic molecules. Here is a not particularly informative illustration of the lysosome. They're referring to the membrane there as a plasma membrane. Actually, it's a cellular membrane. The word plasma membrane is usually restricted to the exterior membrane that holds the whole cell together. But here is a look inside, and there are these clusters of what are called hydrolytic enzymes. These are enzymes that basically use water to tear organic molecules apart. They can tear apart carbohydrates, proteins, lipids, nucleic acids. They can tear apart all manner of organic material. Why would you want to have such things inside your cells? Well, uh, because you might want to take food molecules. So this process of bringing in these food in this manner is referred to as endocytosis. There's some sort of food material, I have no idea what that is, coming in from outside the cell in this process of endocytosis. I told you we would see the word endo more than once. Endo within, cyto cell, bringing it into the cell. So there's this part of the plasma membrane that is pocketing inward, and it pinches off, and you now have what we call a food vacuole right there. The word vacuole just means a little, kind of like a big vessel in a manner of speaking. And the lysosomes will now come along, the lysosome there in purple, and it will now merge with the food vacuole, and this process will now take place in which the enzymes of the lysosome break down the materials inside in the food vacuole. Well, it's now digested. The word for breaking down materials is digestion, if it's taking place inside the cells or inside your stomach. One problem with this illustration is that the final product there should be purple. They're using purple for lysosomes and that sort of yellowish tan for food vacuoles. Once the molecules are broken down, the enzymes are still there, so this is still a lysosome. So it really should be purple. Just a little side note there for you. One of the other things that the lysosomes will do is they will also break down materials that are already within the cell. So for instance, this one lysosome off to the left-hand side is busy breaking down an old, worn-out mitochondria. And we haven't got to the mitochondria just yet. But another organelle, the mitochondria do wear out, and the lysosomes will engulf them and break them down and recycle them. And they will now release them to the outside world, to the cytoplasm. The materials can now be used again. So they break down food and they recycle old materials. Molecules that are. Here's another strange thing that the lysosomes are associated with. Here's a tadpole right there, and the tadpole has no legs, it has no lungs, it's got this fin on its tail, and it's going to develop into a frog, but a frog needs legs and it doesn't need a tail. Tail gets in the way of leaping forward. So it's going to need legs, lungs, and no tail, and eventually that will begin to occur. The legs will sprout off. And the tail fin and the whole of the tail will go away. Well, how does that take place? How do you go from a tadpole with no legs to a tadpole with some legs, and finally, how do you get to that beautiful bullfrog at the end? And the answer is lysosomes. During the developmental stages of the frog, the lysosomes will break open in specific cells and take that cell apart. Just recycle the entire cell and now move that material to other parts of the body. So the entire tail will be taken down. The gills that the tadpole has will be taken down, and that material can be used now to make lungs or legs or whatever it might be. And this is called metamorphosis. Same thing with insects. You get a caterpillar here munching on my plants, and it'll munch and munch until it's stored up enough energy, and then it will form up into a chrysalis. Well, if you were to take this chrysalis and open it up, like about 2 or 3 days after it has been formed, you wouldn't find a caterpillar in there. All you would find would be a bunch of caterpillar sap. All the cells have been broken down with the exception of a few that retain the DNA information for how to build the butterfly that will emerge from this process. So the lysosomes are breaking down the caterpillar cells, and now the material that made up those caterpillar cells can be reorganized into a butterfly. Now, in mammals, we don't call it metamorphosis; we call it development. But there's a an embryo there with its gill bars and its neck fin and its tail and whatnot, and its eye spot. Well, as time goes by, you need to get rid of the tail fin. You need to get rid of the you need to get rid of the neck fin. You need to get rid of the tail. You cannot show up at junior high school with a neck fin. You would be bullied, and so we got to get rid of that. And what will happen is the lysosomes in those cells will break open and tear those cells apart, and now the material can be used to produce new organs, new features, developing into a fetus. And this process continues until you finally wind up as a full-grown human being like Cary Grant here, or depending upon your genetic structure, Salma Hayek. That's the lysosomes there. They're helping in this process of metamorphosis and development. And there are other things that the lysosomes do. They're oftentimes referred to as suicide bags. This is because at a certain point in your lifespan, the lysosomes will just break open. Here's the thing, think about it. The lysosomes have these enzymes that will break down carbohydrates, lipids, and nucleic acids, proteins, everything. Well, if it's breaking down lipids, and if it's a vacuole that has a membrane around it, the structure of the lysosome is that it has a membrane around it, and it has these enzymes within, well, the chemical composition of that membrane is phospholipids. Well, that's a lipid. So how come the enzymes don't tear apart their own membrane and break down those phospholipids? Good question. And the answer is they're fed ATP. They're fed adenosine triphosphate. These enzymes would rather feed upon adenosine triphosphate than phospholipids. So as long as you can keep feeding them, they'll behave and they'll stay inside the lysosome. But as soon as you stop feeding them, they break out of their cage, as it were. And why would you stop feeding them? Because your mitochondria begin to wear out as time goes by. Everything wears out eventually, and your mitochondria are no exception. And the level of ATP that is needed to sustain the lysosomes is the output from the mitochondria drops below that level, and now the lysosomes break open, and they tend to do it all at once, and they take out the entire cell. If one lysosome breaks open, that's not a big deal. The pH inside the lysosome is very low, around 5 or 4.8 or something. The pH inside the cell proper is much higher than that. But when a bunch of lysosomes break open, that changes the pH of the cell. So this is why, as people get older, they tend to shrink up, because they begin to lose, well, they lose bone mass, but they also begin to lose cells. They become smaller, and eventually, and if you get enough cells removed in some critical area of the body, well, you're not going to be around much longer. You're going to be, sitting there with X's over your eyeballs. So they're sometimes referred to as suicide bags, because eventually, if you don't get hit by a bus, or you don't get killed in gangland crossfire, if you don't accidentally fall into a geyser, you wind up being taken apart by the lysosomes. So they're sometimes referred to as suicide bags. Well, the show so far, what have we seen? We have seen the nucleus, the nucleolus, the nuclear pores, endoplasmic reticulum, transport vesicles, Golgi body, secretory vesicles, lysosomes, food vacuoles, and so forth. Which of these are double membraned? Which of them have a membrane around them? Which are made out of simply protein? Which of them have enzymes within? Which of them are involved in breaking down materials? Which of them are involved in transporting materials? And if they do transport materials, what kind of materials are we talking about? Are we talking about finished proteins, unfinished polypeptides? Where are they going? Where are they coming from? Are they coming from the endoplasmic reticulum to the Golgi body, from the Golgi body into the cytoplasm? What are the lysosomes doing? What's the function of the endoplasmic reticulum? It holds ribosomes in place and manufactures transport vesicles. So these are the things that we want to be looking at for the exam. What do these things do? What are they made out of? What's their structure? What's their relationship to one another? So these are organelles that we have looked at so far. We've also looked at other kinds of concepts here. The chromatin, the idea of transcription and translation, the endomembrane system, and endocytosis. So we'll see another little list like this later on, because we're going to continue on with the organelles. We are at about the halfway point in this particular PowerPoint, so if you wanted to take a little break, that would be entirely understandable. So that was a nice little break. We have a couple more membranous organelles to be looking at here. And the one that we're going to look at is that sort of kidney bean shaped object. There it is right there. And this is the organelle that we were looking at just earlier, or we made reference to it earlier. This is the mitochondrion. This is the seat of ATP production. The outside, the mitochondrion, right there. It's another double-membraned organelle. Remember that one of the double-membrane organelles is the nucleus. We'll see another one later on, the chloroplast. A double-membrane organelle. It is the seat of ATP manufacture. Almost all of the adenosine triphosphate manufactured in a typical eukaryotic cell is going to be found here in the mitochondrion, because this is where cellular respiration takes place. There are three major steps in cellular respiration, and two of them take place within the mitochondrion. Cellular respiration produces about 36 ATP, and 34 of them are produced here in the mitochondrion. So a rather important organelle. It is, again, double-membraned. You can see there's sort of a purple area inside. This is what they call the matrix, which means the mother material, and which doesn't make any sense, but that's what they call it. And then you can see that there's this membrane, this sort of zigzaggy membrane surrounding the matrix, and this membrane in the interior there is called the inner membrane, which makes perfect sense. And it's folded up into little pleats, and these pleats are called cristae, which is the Greek word for pleat. Then there's an outer membrane, smooth outer membrane, and all of these features are important in the manufacture of ATP in the cellular respiration process that we will look at. Why is it called the mitochondrion? Well, here's an electron microscope photograph of a mitochondrion. And if you look at it, well, it has kind of a granular appearance, and chondria means granular, so mitochondrion, chondrion means granule, and mito means thread. We'll see the Greek word for thread, mito, later on when we look at mitosis. Greek word for thread, well, the little cristae, the little pleats that you can see there inside the mitochondrion, they kind of looked thread-like to the early microscope workers. So they refer to this thing as the thread granule. And, okay, that's what they call it, the mitochondria. And they didn't know what it did, so they didn't name it after its function. They named it after its appearance. Also, you can see sort of in the upper right-hand corner, there's kind of like a tomato slice there. There you're looking at a cross-section of the mitochondrion. And in between the tomato slice and the mitochondrion, right in the center of the slide, there is what looks like some kind of psychedelic Mickey Mouse with a big long tongue. And I don't have any idea what that's about. But be that as it may, and I'm sure it wasn't, this diagram is looking at the idea that here's a glucose molecule up towards the top of the slide here, and it's entered through the plasma membrane, and it's going into the cytoplasm, and enzymes will immediately begin to break the glucose down and produce a little bit of ATP. And the glucose is broken down to another compound called pyruvic acid or pyruvate. We don't need to worry about that now. We'll see that later in the cellular respiration section. But this breakdown product of the glucose, the food molecule that's coming in, is now inserted into the mitochondria, and various enzymes inside the mitochondria will now take that thing apart. And what we're looking for is we're looking for the electrons that are in the food molecules. We're going to pull these electrons out, and we're going to use these electrons to now, finally, make some sort of usable form of energy. We're going to make ATP there in the matrix, and mostly there in the cristae section. We're going to make a whole bunch of ATP, and that's cellular respiration, and we'll look at the structure of the mitochondria and exactly what's happening in cellular respiration when we look at the cellular respiration section. One last organelle to look at is the smooth endoplasmic reticulum. There it is right there. A series of sort of pipe-like structures coming off of the rough endoplasmic reticulum. It's called the smooth endoplasmic reticulum because there are no ribosomes on it. Smooth pipe-like membrane, and one of the things that the smooth endoplasmic reticulum is doing is it is manufacturing phospholipids. Look at the fact that the smooth endoplasmic reticulum is running into the endoplasmic reticulum. And remember that the endoplasmic reticulum is constantly producing these transport vesicles, and the transport vesicles are going out to the Golgi body. The Golgi body is absorbing the materials in the transport vesicle, not just the polypeptides, but also the phospholipids that make up the membrane of the transport vesicles. And they're incorporating that membrane into the Golgi apparatus membrane, and then they're releasing membrane, these phospholipids, into secretory vesicles, and the secretory vesicles now go out into the plasma membrane. Well, there's no return plan. There's no bottle deposit on the secretory vesicles. They just merge into the plasma membrane. They might be taken down at some point in time, but they're not directly returned to the endoplasmic reticulum. So you have to have a way to replace these phospholipids. You have to have a way to replace the membrane. And there are enzymes inside the smooth endoplasmic reticulum that do exactly that and break them down. So the smooth endoplasmic reticulum, or SER, has no ribosomes on it, but it does have enzymes inside that manufacture phospholipids. And those phospholipids are now transmitted into the endoplasmic reticulum. Now, there's another set of enzymes that are found within the smooth endoplasmic reticulum, and these enzymes help to detoxify the cell. Now, detoxify the cell? Well, yeah, various kinds of toxins can get into the body, and they need to be broken down. And a lot of the enzymes that are responsible for breaking down these toxins are found within the smooth endoplasmic reticulum. So, for instance, take the fairly potent neurotoxin ethanol, also known as alcohol. It enters into the body. It would probably continue to disrupt nerve cell functioning for quite a long time, but it is eventually broken down by enzymes inside the smooth endoplasmic reticulum. So suppose you're a human being about 200,000 years ago. You're moving along, let's say, the east coast of Africa, and you're looking for food, and it's the springtime, it's the summer. You find fruit on the trees. It tastes pretty good. You eat it. Now it's getting into autumn, and food is being it's harder and harder to come by. So, but there's still some fruit on the trees. You eat it. It tastes kind of sour. But you got to eat something, so you eat it, and it doesn't taste too good, but after a while you keep on eating it, and it starts to taste pretty good, and you start to feel pretty good about yourself. You start to laugh at your own jokes and make a fool of yourself at parties. Why is that? Well, because there's ethanol being produced in the fruit. There are bacteria that are converting the sugars in the fruit into ethanol. So it's a common environmental toxin. So we have DNA strands that allow us to break down this toxin. Now, by the way, people vary in their abilities to break down this toxin. Some people don't break it down very well at all. I'm not saying you can drink a bunch of alcohol and don't worry about it. Your smooth endoplasmic reticulum will take care of it. No, people die from acute alcohol poisoning all the time. They also die from cocaine and heroin overdoses. And the smooth endoplasmic reticulum actually has enzymes that will take care of some of those compounds as well, but it can only do so much. Now, if you look at this illustration of the organelles and the ribosomes included, we didn't really talk about the ribosomes quite so much yet. I'll probably mention them in greater detail when we get to the protein synthesis section. But all of these organelles, including the ribosomes, are sort of, well, they're kind of floating around inside the cytoplasm, or are they? No, they're not. They're held in place by these giant green beanstalks that you see going every which way. They're not really giant green beanstalks. They're actually protein molecules. And they form up what is referred to as the cytoskeleton. Well, cyto, as we know, means cell, and the word skeleton means skeleton. So the cell skeleton, the cells have a skeleton. These are all made out of protein. These microtubules, these filaments, they are all made out of protein, and they are holding the organelles in place. Now, think about it. Organisms need to be highly organized. That's why we call them organisms. And you can't be organized if you cannot rely on the positions of the components that comprise you. Let's suppose you have a kitchen. Well, in a kitchen, you're supposed to have a certain organizational pattern. There's supposed to be a certain relationship between the sink and the counters and the oven and the refrigerator and so forth. If you were trying to bake a cake, let's say, and your refrigerator kept moving around the room, or the oven was suddenly up on the ceiling, or the counters were just spinning off into space, you wouldn't get that cake baked. It's not going to happen. You need to have everything fixed, organized, predictable. Same thing with the cell. When the mRNA leaves the nucleus, moves out through that pore, and it's carrying information for how to make insulin, for instance, it needs to find that endoplasmic reticulum immediately. It needs to find ribosomes in a predictable place. The ribosomes that are out in the cytoplasm are not floating freely out in the cytoplasm, as I think the book actually says. They're not. You see these little red balls there on the beanstalks? Well, those are ribosomes. They're held in place so that they can be found. When the transport vesicles leave the endoplasmic reticulum and they head off for the Golgi apparatus, they're not just floating around hoping that they're going to find the Golgi apparatus. No, they're gliding on tracks. I don't know if you've, I don't know if they still have it, but at Disneyland they used to have these canoes, these boats, and they sort of gave you the impression that you would get on these canoes. It was a ride, and you would paddle this canoe. As it turns out, the canoe was on a track, so that limited the liability. But of course, in this age of we can't do anything because of liability fears. They took that ride out. I'm pretty sure it was a fun ride. But the transport vesicles are gliding on cytoskeleton tracks as they move off towards the Golgi body. Now we'll look. Here's another view of the cytoskeleton, holding a mitochondrion in place, holding ribosomes in place. It says that it's holding the endoplasmic reticulum in place. Actually, it looks a little bit more like smooth endoplasmic reticulum, except that it has ribosomes on it. But anyway, various types of elements, microtubules, microfilaments, and so forth, that are holding the organelles in place, sort of like a scaffolding. And the book will go into a certain degree of detail about the microtubules, the intermediate filaments, microfilaments, and so forth. I'm not too worried about that. There are different kinds of cytoskeleton elements that do different tasks. Some are more likely to be holding organelles together. Some of them are more likely to be producing the general overall shape of the cell. So here is what was once a cell, but everything in that cell that was not cytoskeleton was removed. And you can now see the shape of the cell sort of hanging in space there. You can see also where the nucleus was trapped inside the cell. You don't want the nucleus just bobbing around inside the cell. You want to have it in a predictable place. And you can see that the cytoskeleton was doing that. Here's another picture of a cell with nothing but the cytoskeleton. And these are some kind of nifty images of cells that have been stained, different fluorescent dyes, showing different kinds of cytoskeleton proteins. Here's another one here. Looks like a bunch of electric fried eggs or something. And you can see some of these yellow fibers are passing from one cell to another, holding the cells together. Here's now they're sort of stained in red, and you can see the same sort of thing here. There's another one right there. So the cytoskeleton holding the cells together, keeping the organelles in place, attaching one cell to another. They do a lot of different things. Now, aside from all of that, from these obvious structural functions, they also are involved in cell motility. Remember that motile means that you can move from one spot to another under your own power. Well, here's some sperm cells here, off on their nefarious deed, and you have these tails. Well, they're flagella. Well, these flagella are made out of cytoskeleton proteins. One of the things about the cytoskeleton is it, in and of itself, can move. You can take it down and rebuild it, which is sort of like motion, but also you can add ATP to these proteins, and these proteins will move. Here is a protistan, a protistan parasite, a trypanosome, in someone's bloodstream here. It's don't worry, it's just an illustration. And it's got this flagellum. Well, this flagellum is a bunch of microtubules that are encased in the plasma membrane, but if you add ATP to these microtubules, they will change shape, and that will allow the organism, be it a sperm cell or an organism, to move forward, sort of like a little outboard motor. Here's a look at the flagellum right there, all these microtubules that are in these rows here next to one another, and they are connected to one another. Well, they're not necessarily connected, but they can be connected to one another by yet smaller proteins called dynein arms. Well, when you add ATP to these dynein arms, they twitch, and that causes them to move the microtubules next to them, and that causes sort of an undulating movement down the flagellum, which allows the organism to now be moved forward, or the cell, in the case of a single sperm cell, to be moved forward. So you have these long microtubules that can be ATP activated and will now wiggle, and the cell can now move through a fluid medium. So that's the flagellum there. The word flagellum actually it means whip. I might have said that at some point in time. This is a flagellum on a sperm cell, and you can see that they're showing the flagellum. They're actually sort of mismarking it a bit. The microtubules would be the blue structures beneath the plasma membrane, but the plasma membrane that surrounds the microtubules would actually be considered to be part of the flagellum. So you can whip that flagellum back and forth and move forward in a fluid medium, like water, for instance. Now, some other organisms, they don't have flagellum, they have cilia. Cilia right there. The word cilia means little hairs, and you can see these sort of fuzzy little hairs surrounding this paramecium here. Cilia means little hairs. The singular form is cilium. What's the difference between a cilium and a flagellum? Well, the length. That's it. They have the exact same microtubule structure in the cilium that you have in the flagellum. It's just that the cilia are shorter. So why would you want them to be shorter? You might want them to be shorter because you might want to move with greater dexterity. If you have a flagellum, it's like having a very big outboard motor on a boat, just sort of launching you forward, allowing you to sort of wiggle forward. And they don't have a lot of accuracy. They just sort of go kind of forward. They go in that direction. But with cilia, you can move each cilium by itself, or you can move whole fields of cilia. You can spin in one place. You can rise up. You can pop back down. You can navigate through little crevices and so forth. It gives you a much greater degree of precision in terms of where you're going to be going. But it's the exact same idea. You have microtubules that are activated by ATP, and you can wiggle them, and now you can move from one place to another. Now, if the cell is in a fluid medium, like water, you can move through the fluid medium. But if the cell is held down, if it's stationary, if it's held in one place, then the cilia will not move the cell through the water, but they will move water over the cell. Now, we actually have cilia in the human body. So, for instance, in the human female reproductive tract, there in the fallopian tube, which arises from the uterus, you have cilia. Why so? Well, here's the idea. There's the ovary there. The ovary is going to release an egg, and this egg is now going to be released into the pelvic cavity. Well, you can see the sperm cells that are moving down the fallopian tube, and they're heading towards an egg, which is in fact in the fallopian tube, and that's where fertilization takes place, there in the fallopian tube. Problem is, the fallopian tube, as you notice here, does not directly connect to the ovary. So there is every possibility that a given egg could be released, and that egg might never make it to the fallopian tube. It'll simply drift off into the pelvic cavity. Here's the problem. That does not bother the sperm cells at all. Left, you can see where the nucleus. The sperm cells have one mission and one mission only, and that is to find that egg, and they will keep on looking for it. They will fertilize that egg, or at least one of them will, out in the pelvic cavity, and that's not good because now you have a developing embryo in the pelvic cavity. Unfortunately, all of the capillary beds, all of the blood supply, the oxygen, the food that the embryo will need is to be found in the uterus. Which is why the fallopian tube is there, to carry the egg into the uterus. Well, how do you get around this difficulty when there's no actual connection of the fallopian tube to the ovary? The answer is that inside the lining of the fallopian tube, nucleus used to be, you find cilia, and they wiggle. They wiggle back and forth. Here's another picture of some cilia stained with a different kind of dye. They wiggle back and forth, and by wiggling back and forth, they create a current, and now that egg can be sort of wafted into that current and pulled into the fallopian tube and actually pushed along by the cilia, where the fallopian tube narrows there. It can actually be pushed along, sort of like people at a baseball game. Remember when they used to have baseball games? In a stadium, moving batting a beach ball around. So the cilia are designed to correct this somewhat significant design error in the reproductive tract. Now, we also find cilia in the human lungs, in the bronchial tracts that lead into the lungs. Here's the thing, you're constantly breathing in air, and this air has pollen, it has dust, smoke particles, all sorts of stuff in there, and your lungs would quickly fill up with this debris, but on the surface of your bronchi, those little tubes leading into the lungs, you have mucus. Well, mucus is a short-chain polypeptide that's moist, and it'll trap the dust particles. Well, that would now just mean that you would have dirty mucus draining into your lungs, but instead, on the lining of the lungs, you have cilia. Here's a microscopic view of them. You can see that little red fringe up towards the top. Those are cilia, and there they are again. And here's an even closer view, like so. And the cilia sit there, and they sweep the mucus back up out of the lungs. You can now evacuate it into the gastrointestinal tract, or you can spit it out, I guess. So that mucus is keeping your lungs clean. Now, if you happen to be a smoker, you might want to consider the following. Here are the lungs of two human beings, the white one on the left, a non-smoker, and then the sort of brownish-orange one on the right, a smoker. Now, okay, first of all, sure, okay, they're both dead. Got that. But the non-smoker probably died a relatively peaceful death, maybe in the middle of the night, just sort of slipped away. The smoker died a terrible hacking death, coughing up pieces of his or her own lung. Why is that? Because when you smoke cigarettes, the regular tobacco cigarettes, tobacco contains all sorts of plant tars and other particles and just regular smoke. And the tars bond onto the cilia and keep them from doing their job. So you're simultaneously knocking out the defense system for smoke and other particles while simultaneously inhaling all sorts of smoke and particles. Every once in a while, someone will ask me, is it the same thing with marijuana? Yes, it's the same thing with marijuana, but usually people don't smoke 2 packs of marijuana a day. Is it the same thing with the vaping? Not that I know of. I don't think you have the same particle load in with vaping. So the cytoskeleton is responsible for these flagella and for these cilia, but it also does something else. We kind of saw this earlier. It creates the shape of cells. We're looking at this this view of an animal cell, this sort of abstract view of an animal cell, and it has this sort of amorphous shape to it. Okay, well, actually, most animal cells have definite shapes to them, and they have definite surface features that are provided by the cytoskeleton. This right here might look like cilia, but they are not cilia. These are referred to as villi. Now we're looking at a cross-section of a human intestine, and these villi are rising up off of the surface of the intestine, and they're reaching out into the space into the intestine where food molecules are now passing through. It kind of looks like shag carpet. Now, if you don't know what shag carpet is, you're not missing anything. It was this terrible kind of carpet that they used to have, especially in the 70s. And it had these long threads that would stick out in this manner. And basically it would just trap all kinds of food and other particles and so forth. You had to like shampoo them on a regular basis. It was just awful. So that's with the shag carpeting, having these projections, that's a bad thing. But in your intestines, it's a good thing because it's increasing the surface area to absorb food. You want to absorb food particles in your intestines, good thing. The floor of your living room, a bad thing. So these villi are created by the microtubules. You can see the microtubules, these long sort of thread-like things that are pushing out the plasma membrane, creating additional surface area. If you were to remove the villi, you would just have one flat surface area to absorb the food. You wouldn't be able to absorb enough food. But because the villi are being pushed out here, you have, I haven't even calculated it. It's going to be probably about, I don't know, at least 10 times the amount of surface area just in this one plane right here, more than that. To absorb food. You need it for the intestinal cells to do their job. How do they do their job? Because the cytoskeleton gives them the shape to do their job. Here are some nerve cells, some neurons, and you can see they're kind of long and thin. Actually, they're not drawn to scale. The typical nerve cell at this scale would be about, I don't know, hundreds of yards long. Nerve cells, in order to do their job, to transmit electronic information along their length, they need to be long and thin, sort of like telephone wires or internet cable wires. They need to be very, very long and thin. If they were round, they wouldn't be able to transmit information in any particular direction. Because they're long and thin, they can do so. One thing you might also notice here on this neuron, you can see that the neuron is coated by what they're referring to as myelin. That is that cholesterol coating around the neurons that holds the electromagnetic information in place. But if you want to send a message along a neuron, the neuron has to be long and thin. How do you get it to be long and thin? You arrange the cytoskeleton to make it be long and thin. The other thing is that when one neuron attaches to another neuron, they attach at very, very narrow junctures known as synapses. And one neuron, right here, the sort of bell-shaped ending of one neuron, must attach very, very closely to another neuron. It can't be floating off in space somewhere else. It needs to be right up against that next neuron. And strangely enough, the neurons do not connect to one another electrically. They actually, the message, the electrical message is carried along the neuron. And when you get to the end of the neuron, it's released as a chemical message. So you have these little red dots here, which are neurotransmitters, and they now go across the synaptic cleft, that synaptic gap, and they now stimulate the next nerve cell and a new electrical discharge is produced. But you have to get the two neurons together just like so, very, very close to one another. And if they're not close to one another, then it's not going to work. They're not going to be able to carry a message from one nerve cell to another. They have to be very, very close. Well, how do you get them to be close like this, and how do you get them to stay close like this? And the answer is, if you, how many people are thinking cytoskeleton? Well, if you are, you're thinking right. Cytoskeleton elements, little proteins, will knit the neurons together. Here's the thing about thinking. You have to have your cytoskeleton connecting your neurons in order to think of things or remember things. Let us say that you're going to go ahead and you're going to memorize the state capitals of the United States. And you know, for instance, that Austin is the capital of Texas. Let's say you didn't know that before, but now you're building this new information in your head. You know that you're told that Austin is the capital of Texas, that Baton Rouge is the capital of Louisiana, and so forth. Well, you have to have a neural pathway, a series of connected neurons that allow you to think what a state is, what a capital is. And you have to have these pathways connected to one another. Now you have to have a neural pathway that will allow you to think of the word Texas. And now you're told that the capital of Texas is Austin. Now you have to build a new pathway for Austin and connect it to these other pathways. And if you never knew what the state capitals were, and someone told you, well, Austin is the capital of Texas, and they were to ask you again a month later, and you hadn't thought about it, you probably wouldn't remember. And that's because on the day that you first learned it, you built this new pathway. You connected a bunch of neurons using cytoskeleton, but if you're not using that pathway, the brain, the body will take that pathway down and use the cytoskeleton for something else. And it goes away. It's as if you never knew it to begin with. So you have to build that new pathway, and then you have to think about it. You have to think about it later on that day, maybe several times the first time that you learned it. And you have to think about it later on, and think about it later on yet again. Every time you think about it, the cytoskeleton elements become more and more embedded, and you are more able to remember it. And now maybe it flees your memory for a while, but as long as the cytoskeleton is still there, you can retrieve it in the future. Now, the information is still in your head. It's still being held in place by cytoskeleton. This is why it is so difficult to cram for an exam. Because you have to build so many cytoskeleton elements so quickly, it wears out the body. It becomes beyond the capacity of the neurons. Let's suppose that you were going to enter a weightlifting contest, and it's going to be 3 months from now. Would you wait until the day before the weightlifting contest to start training for it? Of course not. You'd start right now, and you'd ease into it and go at a proper pace. But you would be building up muscle cells. You'd be building up the proteins and the glycogen inside the muscles. That takes time. It's the same thing with learning things. It takes time. You have to build these connections, and then you have to reinforce those connections, and then you have to be able to access those connections. And a lot of this work, not all of it, but a lot of that work, is done by cytoskeleton. Well, speaking of muscle cells, muscle cells are also have cytoskeleton elements within them. We've mentioned them several times. Actin and myosin are essentially cytoskeleton elements, and they connect to one another, and they're shown here in this diagram, the myosin in red and the actin in blue. You add ATP to them, and they slide across one another. That allows the muscle to contract. So essentially, the cytoskeleton allows cells to move about by means of cilia and flagella. It also allows muscle cells to contract, which allows the entire organism, the entire animal, to move about. So the cytoskeleton does a lot of things. So the show so far, we've seen the mitochondrion, the smooth endoplasmic reticulum, cytoskeleton, flagellum, cilia, and so forth. Again, you want to know what these things are made out of, what they do, what their function is, and so forth. So let's look at the learning objectives for Unit 8. The ones that we looked at here in Part A focus on animal cells. There's actually only one learning objective for this section, Unit 8, Part A, but it's a pretty big learning objective. It says be able to identify the functions, what does it do, composition, what is it made out of, and structural characteristics. Are they double membraned? Are they just a particle, a protein particle like the ribosomes? The functions, composition, structural characteristics of the organelles of eukaryotic cells, including the nucleus, the nucleolus, endoplasmic reticulum, ribosomes, Golgi apparatus, transport vesicles, secretory vesicles, etc. It says etc., etc. at the end. Well, we mentioned things like the endomembrane system. We looked at the concept of the fact that the DNA is involved in the transcription process. The ribosomes are involved in the translation process. We'll look at that kind of stuff in a little bit more detail later. But this is essentially the list here of the cell features, the organelles, that we have been looking at. So you want to know about them. You want to know their functions, composition, structural characteristics, and other things that might have been mentioned here and there. So here in Unit 8, Part A, we've been looking at eukaryotic cells, and we've been focusing on animal cells. Well, when we go on to Part B, we will still be looking at eukaryotic cells, but we're going to look at plant cells. Plant cells have essentially almost all of the organelles that you find in an animal cell. There's one organelle in the animal cell we didn't really mention at all. That's the centriole, but we'll mention it in the plant section. Plants have 3 more major organelles that we will look at in Part B. Cell attached to other cells. This is an animal cell. We're focusing mostly on animal cells here in this section. And, well, the animal cells don't have cell walls, so how do they attach to one another? Well, with plants, there's basically glue that will hold the cell walls together. You can't do that with animals. The cytoskeleton is holding the cells together. The cytoskeleton will reach out from one cell to another and connect the 2 cells. If we were to remove the cytoskeleton from your body, not only would all of your organelles now be completely mixed up and confused, but every cell in your body would basically, well, slurp to the ground, and there would be this sort of a cellular soup there, and there'd be a bunch of tendons, ligaments, and bones that would be all that would remain. So you're being held together by cytoskeleton. The outer plasma membrane later on, but and some food materials will move through this plasma membrane, but some can't. So they're taken in by these food vacuoles, and the lysosomes will now merge with these food vacuoles and break down the food molecules that are coming in. Well, the food molecules are carbohydrates or proteins or lipids, whatever they might be, and the lysosome can break them down and now release them to the cell, and now you can use the energy in them to make ATP or to manufacture new materials.